Hi, I'm Marisa Torres. I'm a Bioinformatics Group Leader at Livermore National Lab, and I'm also the WIDS Livermore Ambassador. Hi, I miss you all. And today I'll be sharing with you about how I'm killing diseases with really big computers. So we're building analysis tools to solve disease. I do bioinformatics for experimental design, simulation, and prediction. And some of the projects I'll share with you today are ATOM, the American Heart Association, and our work on COVID. And by the end of this talk, I want you to understand how we're using data science and supercomputing to help us better understand how drugs and diseases affect a human. First, I'll talk about ATOM. This is a consortium between national labs, academia, and industry, and we're using machine learning to speed up drug discovery, accelerating therapeutics for opportunities in medicine. And our goal is to deliver treatments to patients within a year of identifying possible drug targets. We've built the ATOM modeling pipeline AMPL to make compound property predictions with experimental data modeling. Our core functionality includes data libraries, machine learning models, and simulations and processing. And we run several of our large predictive biology capabilities using this. So let's look at some of the details. I'll spend some time talking through these arrows and spending some equal attention on the smaller arrows, which represent our relevant predictions for downstream analysis. With ATOM and our modeling pipeline, we need to find new treatments very quickly. So we built the generative molecular design loop. And we're using GMD to be able to identify drugs for repurposing for cancer treatments, as well as finding new drugs for cancer and antiviral treatments. Turns out we never have enough data, and so we're using machine learning and simulations to help fill in the gaps. So we're building up our data libraries. And we start with fundamental data pre-processing. So we start with assay activities as well as potential drug compounds. Then we do a featurization of these SMILE properties where we go from compounds, featurize them using their SMILE strings into molecular learning descriptors. We also featurize assay activities, and so these are key activities for drug design: pharmacokinetics, safety, and efficacy. Really big computers. Livermore's capabilities are not only on the top 500 list, but they're a specialized collection of machines, several of which are highly ranked. And we're finding the right balance for our workload. For example, we need lots of memory to be able to do database searching, or lots of CPU cores for tasks like docking simulations, and then GPUs. They help us do model training. So we train these AMPL activity models so that we can better search our potential drugs for these key drug design activity models. And we use a machine called Corona, and this has 14,000 cores and over 200 GPUs. So we run these predictions to be able to get assay activities. We start with our 2D SMILE strings, and we run them through our activity models, and we get both predicted assay activities as well as developability of these SMILEs. So these predicted activities, they let us flag potential drug safety issues. We want to know if a compound to protein pairing is meaningful. We want to know if we're binding to potential safety categories or to non-targets. So we know for 6,000 known drugs, can we predict whether we're binding to these AMPL safety categories? Also, for 1,600 known drug targets, we want to know whether we might be getting potential off-target binding. So moving on, thinking about how a drug is affecting a human, we found that we really had to start simulating some of this data. So we partnered with the American Heart Association. These safety and off-target questions are also very important to them. And our goal with this partnership is to be able to scale up our drug analysis. We're scaling up our simulation capabilities. We're also better predicting protein binding as well as protein docking sites. So we started with creating these fusion models, and this provides us a way to better use our structure data to predict protein binding. Pictured here is a 3D protein model with a drug in a predicted binding site. So we're featurizing PDB bind. This is a specialized database that has 3D complexes with binding activity. And we featurize these to create 3D complex properties. We then train these fusion models, taking as input these 3D complex properties. And then in 2020, we were interrupted by our work in the COVID-19 pandemic response. So why is Livermore able to help here? We've long been a part of the nation's pandemic rapid response, and our teams are veterans of both the BioWatch and BASIS pathogen detection programs. So we started running our AMPL and our simulations to be able to understand which compounds might have correlation with experimental data. So now we're predicting to COVID proteins. We start by making 3D protein models using our homology modeling, AS2TS, and then we identify docking sites, starting with potential drugs. Using PDB spheres, we identify these protein docking sites. And we're running these on a machine called Quartz, which has over 108,000 cores, and Mammoth, which has 8,000 cores and 131 terabytes of RAM. So now we're running these large-scale docking simulations to be able to fill in some of the data we need for our machine learning. Two of the docking approaches are conveyor LC and mechanistic 3D modeling, GBSA. And this is using a machine called Lassen, which has 34,000 cores and 3,000 GPUs, and Ruby, which has 84,000 cores. This lets us predict protein binding through these docking simulations. And we run docking and taking as input the 3D protein models and docking sites we identified, as well as potential drugs. And we output predicted protein binding and these protein compound complexes. Now we have the input to be able to predict protein binding using those fusion models and also using 3D complex properties. As an outcome of using simulations, we were able to create an entirely new COVID-19 drug efficacy category using predicted protein binding down-selected from our simulations. This lets us create an entirely new category that's not based on the assay activities that the other categories are based on. We screen compounds against two SARS-CoV-2 proteins, Spike and MPRO. We then down-selected through the processes that I described for experiments, docking, GBSA, efficacy, and fusion. We identified 10 compounds that are potential antivirals. We screened several hundred of these, and we found 5 promising compounds that were strongly inhibiting in vitro. And these are the predicted docking poses that we now have as part of our data set. We released a tool called CoViewer within months. The team received the DOE Secretary of Energy Achievement Award. And this CoViewer tool helps COVID-19 researchers explore our molecular dynamic simulations. Back to the American Heart Association partnership. We're now scaling from the 4 COVID proteins now up to thousands of human proteins. So now that the data is organized and searchable, what can we do? We want to start looking at the known drug and target pairings. We also want to look at what we can confirm experimentally. And we're starting to identify the gaps where more experimental data would be helpful. We have these huge observation sets with many opportunities to investigate. For example, in the safety category, we have 28,000 predicted assay activities. We also have 24,000 predicted protein bindings. And from these 11,000 3D modeled proteins, only 14% are known drug targets. And for the remainder, we don't yet know what to do if we see some off targets. And of these known targets, we have made 3.8 million off-target binding observations. Turns out we only know targets for 3,700 of the known drugs. So this leaves us with a very small set of possible pairings we can confirm are on target. So let's start searching for cardiotoxicity or liver safety issues. This graphic is from the Mamashina paper, where they looked at mechanisms of drug-induced cardiotoxicity. And they're trying to see which drugs actually cause issues. And 78 drugs are known to have cardiac adverse effects. So we started investigating 18 of these drugs that overlap to our AMPL safety category. So are our models adding value beyond their input data? Here's an example of 4 drugs with AMPL data, where we're seeing both docking and AMPL results that are matching the published toxicity mechanisms. And we're starting to connect the safety activities with the protein binding. 18 of these published drugs are linked to AMPL, and these activities are associated with 3 cardiotox proteins, which are pictured on the left, and 1 liver protein, which is pictured on the right. And we can see some of these drugs are binding to the proteins. This is an example for loperamide, where 2 proteins have docking results. So we want to be able to confirm our predictions and compare them to evidence from a spoke precision medicine graph. And here we can see that we are seeing matching experimental results to our predictions. Our AMPL and docking observations are adding value. We're starting to fill in the gaps. And again, using loperamide as an example, without AMPL and docking, we would only know about these experimental matches. With these predictive biology capabilities in place, we're now using the generative molecular design in ADAM to be able to find new drugs for cancer and antiviral treatments. And we're continuing to add to ADAM with a new compound design tool, starting from the predicted assay binding and protein binding activities. We are now training our compound variation tools to be able to generate new compounds. And we're designing new AI-based drug compounds. Another related work, the vaccine design project with GUIDE. So a reasonable person might ask, isn't vaccine design the same as drug design? No, actually. For vaccine design, you're actually looking at mutations in the protein space. But we're starting to see for the GMD loop that there is overlap where we could screen compounds to avoid costly simulations and wet lab experiments. So from now on, we get to start with everything here, and we're looking for the next problem to solve. So that's the end. So I'll credit my collaborators until they kick me off the stage. So thank you to Felice Lightstone and Jim Brazzi and all my team members from ADAM, as well as the American Heart Association.
You still have 40 seconds.
Oh, still have 40 seconds?
Yeah, go on. You can continue.
Any questions? Oh, and the team for Livermore Computing, as well as my partners in the DOE NVBL and our LDRD. We have a lot of experimenters we're working with, and statisticians and data scientists, and a lot of bioinformatics expertise.
8 more seconds.
And you can see we have a lot of partnerships with other DOE labs.
Done. Well done, Marisa. Thanks so much.